Chapter 1, Topic 1, How do antimicrobials work? Now, this topic may take the award for having the most objectives. There's a lot to cover in this topic, but let's go over and cover the objectives with you. Be able to state the main goal of antimicrobial treatment, be able to identify the sources of most currently used antimicrobials, be able to describe two methods for testing antimicrobial susceptibility, be able to define therapeutic index and identify whether a high or a low index is preferable, be able to explain the concept of selective toxicity, be able to list the 5 major targets of an antimicrobial agent, be able to identify which categories of drugs are most selectively toxic and why, be able to distinguish between broad-spectrum and narrow-spectrum antimicrobials and explain the significance of the distinction, be able to identify the microbes against which various penicillins are effective, be able to explain the significance of beta-lactamases, be able to identify 2 antimicrobials that act by inhibiting protein synthesis, be able to explain how drugs targeting folic acid synthesis work, be able to identify one example of a fluoroquinolone, be able to name a drug that targets the cellular membrane, be able to discuss how treatment of biofilm infections differs from that of non-biofilm infections, be able to name the 4 main categories of antifungal agents, be able to explain why antiprotozoal and antihelminthic drugs are likely to be more toxic than antibacterial drugs, and be able to list the 3 main targets of action of antiviral drugs. We've got quite a few things to cover in this topic. They are principles of antimicrobial therapy, types of chemotherapy, identifying infectious agents, determining drug susceptibility, how to choose a drug, mechanisms of drug action, examples of drugs, spectrum of activity, treating eukaryotic microbial infections, and treating viral infections. It's hard for us to believe the medications that we know and often take for granted were discovered at the earliest in the 1930s. These medications revolutionized how we treat infections, and the discovery of penicillin prevented thousands to hundreds of thousands of deaths in World War II alone. A lot of these products are extracted and purified from other organisms. For example, penicillin comes from the Penicillium fungi. These organisms produce these antimicrobials as a way to prevent other microbes from invading their territory. An important term we need to discuss here is chemotherapy. We think of chemotherapy as a type of treatment used only to treat cancers, but the term is actually a broader term for use of any chemical to treat any disease. When developing a drug for use as an antimicrobial, there are some considerations that need to be taken into account. You can see these here on the slide. All of these are equally important aspects that need to be measured. There are several types of chemotherapy drugs. These are based on what their intended outcomes are. You can see the major categories on this slide. Take a moment to look through these. 2 important types are narrow spectrum and broad spectrum. This is referring to the range of effect the drug may have. A broad spectrum antibiotic can work against a wide variety of microbes, while the narrow spectrum works on a smaller group of microbes. Think of this as a sniper versus a shotgun approach. However, there are 3 important questions that must be addressed before prescribing an antimicrobial therapy. First of these is, what is causing or likely causing the infection? For example, a majority of sinus infections are viral in nature. This will change what, if any, drugs should be used. After the agent of infection has been determined, the next question is, what kind of drugs is it sensitive to? If you know you have a MRSA infection on your hand, using methicillin won't do any good. Lastly, you need to consider how the patient is doing. If they are in critical condition, you don't have time to root out the infection, while if they are not terribly ill, you have more time to collect information or to see if the infection clears on its own. So, how do we identify the agent? You've been doing a lot of these assays in the laboratory this semester. Some of these are used in the clinical lab. To begin running these assays, samples are taken from blood, stool, sputum, or swabs. Oftentimes, multiple types of samples are taken because it is hard to extract live microbes from all samples. So the greater the variety, the greater the chance of being able to identify the infectious agent. As you can tell from lab, these tests can take several days to a week. Given this information, practitioners will oftentimes start treating based on a best guess. Once we have identified the agent, how do we test for susceptibility? There are two main assays used for this, both of which you should be exposed to in lab this term. The first is the Kirby-Bauer test. You can see an example of this on the slide here. The identified agent is streaked onto an agar plate in such a way as to create a field of growth. Discs that have been soaked in antibiotics are placed on this plate, and the area where growth is inhibited is measured. Here's what you need to be careful of. By looking at the plate, you would automatically assume that enterofloxacin, right here, is the best choice because it has the widest zone of inhibition. This is not quite true because each antibiotic has a different range and susceptibility. For example, some antibiotics may require a zone of 30 millimeters or more to be susceptible, while others only require 17 millimeters. So, for these types of assays, such as Kirby-Bauer, we always have a chart that you refer to that lets you know what the zone needs to be to indicate susceptibility. The other type of assay is known as a tube dilution. Using this assay, the minimum inhibitory concentration, or MIC, can be determined. In this test, the antibiotic is diluted across several tubes with a constant amount of agent added to the tubes. This assay may be more sensitive than Kirby-Bauer, but does require a lot more setup. Now that we know the agent causing the infection and what it is susceptible to, you would imagine that the final selection is easy. That isn't quite the case. There are a variety of facts that need to be evaluated. The first is the therapeutic index. This is the difference between effective dose and the toxic dose. For example, if taking one aspirin would clear up your headache, but 2 aspirins would kill you, this would be a very dangerous drug with a high TI, or therapeutic index. Luckily, aspirin and other over-the-counter drugs typically have a low TI, so it is difficult to reach toxic levels. Besides TI, the healthcare professional must take into account what the site of infection is, what the patient's drug history is, and any other physical factors that may change the way a drug works. Just like we talked about in Chapter 9, it is important for antimicrobial drugs to target parts of the bacterial cells that are different from the eukaryotic cells of the host. This difference is known as selective toxicity. The importance of not further damaging the host cells is critically important and plays a role in the antimicrobials used for eukaryotic microbes and viruses. But we will get to that later. First, let's take a look at the major targets for bacterial infections. I expect you to understand these targets and how they are targeted. Let's look at each of these sites listed above. So, there are some antimicrobials that inhibit protein synthesis by acting on ribosomes. This is possible because bacteria have different sized and shaped ribosomes. So, there's some that affect the large subunit or the 50S subunit. There are some that attack the 30S or small subunit. Some block both. There are some that block the product of a metabolic pathway that eukaryotic cells don't have. An example, or that human cells more accurately don't have. Folic acid synthesis in the cytoplasm would be one of these. Humans are unable to make folic acid, and we have to ingest it. Bacteria, on the other hand, most of them can make it. Cell wall inhibitors. Humans do not have cell walls, so antimicrobials that attack cell walls will generally leave our cells alone. Cell membranes a little more dicey because most cell membranes are very, very similar between prokaryotes and eukaryotes and between different types of eukaryotes, but we do have a few that attack specific parts of the cell membrane. DNA and RNA, there are some antimicrobials that inhibit enzymes that are specific to that organism. For example, there are some that inhibit replication and translation. One of them inhibits gyrase, okay, and they inhibit a type of gyrase or helicase that we humans don't have. There's others that inhibit RNA polymerase that's unique to acid-fast bacteria. The take-home message of these targets is the more different these targets are from the host cell, such as targeting peptidoglycan, the less toxic the drugs will be to the host cells. One of the most toxic targets is the cell membrane, since they are so similar to ours. The next 2 slides has a table that lists specific drugs and their metabolic targets. I don't expect you to memorize this table by any means. Instead, I want you to focus on some main examples that I have underlined. For example, penicillins are one of the more commonly used antimicrobial. As you can note from this chart, they target the cell wall and have a wide range of targets depending upon which subgroup you're talking about. These subgroups of penicillin were manufactured to try to expand the repertoire of target microbes. As you can see, we have been successful at making a variety of subgroups that target different bacterial groups. Now take a moment and look through the previous table and this table. Make sure that you know the ones that I have underlined. You should recognize them already. They've probably been prescribed to you or somebody that you know. Or you may have heard about them in the news, such as, on the previous slide, methicillin. Methicillin-resistant Staphylococcus aureus is causing us a big problem, so that's why I have it underlined. Earlier in this topic, we defined broad spectrum and narrow spectrum. This table easily depicts examples of each of these targets. As you can see, tabramycin or polymyxin are narrow spectrum antibiotics, while tetracyclines has the broadest range of antibiotic effect. There are benefits to each of these types of drugs. If we know that we are dealing with a case of Salmonella osis, we can specifically target this organ without damaging the normal microbiota too much. However, if we don't know what we are dealing with, a broad-range antibiotic can quickly begin to treat the infection, if the target is within the repertoire of targets. Since we are talking about targets, let's talk about biofilms. We used to believe that this had to do with the glycocalyx, but we now believe that the cells involved in the biofilm actually express different phenotypes than their non-biofilm cousins. This can change whether or not a drug will work against it. If you think about how some infections are spread, especially those from open wounds, imagine the implications of this to the healthcare professionals. To combat these specialized infections, we have found a few key methodologies. One of these is to couple DNA ACEs with antibiotics to increase their effectiveness. Another solution is to coat the items to be added to your body with antibiotics prior to insertion. Almost all of the drugs we have talked about already are completely ineffective at targeting yeast, protozoa, and helminths. This is because with the other antimicrobials we are targeting structures that aren't in eukaryotic cells. This makes them ineffective against all of these groups of microbes. While your textbook lists several common drugs that are used for these types of infections, mainly I want you to focus on the issues associated with treating these infections instead of the specific drugs that are used, unless, of course, I have it underlined on the previous tables. Our next most difficult to treat infection are viral infections. As you can imagine, this has a lot to do with the way viruses replicate, and there are very few targets in the viral structure that can be targeted. There are, however, 3 targets we can use in viruses. The first of these is blocking viral entry into the cell. This can be accomplished through blocking the lock and key mechanism or fusion of the virus to the host cell. A second target is the inhibition of the nucleic acid synthesis. This is accomplished through the use of nitrogenous base analogs that terminate in the replication process. Lastly, we target viral assembly or release from the host cell. This can prevent the further spread of the virus. We can do this by using proteases that break down the proteins under production. Well, that's it for this topic, and I've got all of our learning objectives here. Let's go over them. Be able to state the main goal of antimicrobial treatment, be able to identify the sources for most currently used antimicrobials, be able to describe 2 methods for testing antimicrobial susceptibility, be able to define therapeutic index and identify whether a high or a low index is preferable, be able to explain the concept of selective toxicity, be able to list the 5 major targets of antimicrobial agents, be able to identify which categories of drugs are most selectively toxic and why, be able to distinguish between broad spectrum and narrow spectrum antimicrobials, and explain the significance of the distinction, be able to identify the microbes against which various penicillins are effective, be able to explain the significance of beta-lactamases, be able to identify 2 antimicrobials that act by inhibiting protein synthesis, be able to explain how drugs targeting folic acid synthesis work, be able to identify 1 example of a fluoroquinolone, I've got that one underlined, be able to name a drug that targets the cellular membrane, also have that one underlined, be able to discuss how treatment of biofilm infections differs from that of non-biofilm infections, be able to name the 4 main categories of antifungal agents, be able to explain why antiprotozoal and antihelminthic drugs are likely to be more toxic than antibacterial drugs, and be able to list the 3 major targets of action of antiviral drugs.